So first I'm going to lay down, so this is a pipette. It's used for measuring really small quantities of liquid. So I'm taking 5 microliters. So this is loading dye, and because all the fluids are clear, it just helps me see the liquid when I put it in the gel. Okay, so that's about a microliter each of loading dye. That's enough. So I'm going to load 3 microliters, of each sample. So I'm gonna pull up 3 microliters. It's in there. Now I'm gonna mix it with the dye. Now I'm gonna load it in the gel. Loading it into the lane. Yeah, this is a skill. It takes a little bit of time to learn. The first time I learned it, back when I was an undergrad, I poked this sharp little point right through the gel every single time. So it does take a little practice. Okay, those are the 4 samples. We need one more thing. So they'll run out on the gel, and we need to have some idea of how long those DNA fragments are. And so in order to figure that out, we put a sort of a standard right next to it. And this is called a ladder, and it has known fragment lengths of, say, 2,000 base pairs, 1,000 base pairs, 5,000 base pairs of DNA. And so we can measure how long that fragment is that I amplified in relation to this standard or so-called ladder. So I'm gonna put this in the last well. Okay, now I'm gonna put in some buffer. So this is essentially just pure water with various salts in there, because electricity does not conduct in pure water very well. So I'm loading some buffer. And so we have to make sure that the red is here. It runs to red. That's how we remember that it goes to this pole. Now to make sure it's running, you have to look here really closely. And there is a wire that runs down here, and there should be teeny little bubbles coming out of it, and there are. So we know that it's actually working. So we'll come back in 25 minutes. Okay, here we are 25 minutes later, and it's the moment of truth. Here's our gel. So you can see that that blue dye that I loaded up there is now partway down the gel, indicating that it seems to have worked. So the DNA will have run. And the smaller it is, the farther down it will be this way, because it can move faster. And the larger it is, the closer it'll be to the place where I loaded it on the gel. I will grab this tray that transmits UV light, put my little gel fragment on there, get rid of the bubbles. And there we go. Okay, so now we need to visualize it with ultraviolet light.